Alrighty, this is probably going to need to be a couple videos as well. So the first video is going to be smell and taste, and we might do some other things. Okay, so first let's talk about taste. And let's hope that this doesn't die. So you have your cute little tongue, and all along your tongue you've got these little bumps, and then these bumps have taste buds. And these taste buds have neurons that then go back to the brain and tell you, hey, this is what you're tasting. So this is what the taste buds are going to look like. They're each going to be cute little boxes. Let's see if this will make a box, please. Okay, so they've got a couple boxes. Alright, and these boxes have a channel called ENaC. Okay, this one we'll start with is salt. Okay, so you've got some salty taste. So salt is going to come through the ENaC channel. Okay, that's going to send a positive BWONG action potential through. Okay, and it's going to open a channel, your voltage gated calcium channels. The calcium is then going to rush in. It's going to bind to the snare proteins. And then you've got the exocytosis going to your brain saying, hey, we've eaten something salty. Okay, the next channel, or the next flavor, I guess, is going to be sour. Come on. Hello. Please make a square. Thank you. Okay, the next one is going to be sour. So it's actually still the ENaC channel. Okay, so you still have the ENaC channel, but this time when you're tasting something sour, it's the pH that's coming, that kind of like acidic, like sourness, right? And so you're going to have hydrogen come in the ENaC channel. It's going to send that positive charge. And what this is going to do is it's actually going to send a signal to block the voltage-gated potassium channels. So that's going to then increase your voltage. You're going to open your voltage-gated calcium channel. It's going to bind to the snare proteins. And then you've got, hey, what you're tasting is sour. The next one is going to be three things. It's going to be your sweet, bitter, and umami. Okay, so what's going to happen is you're going to have a G protein, which is then going to go and it's going to activate phospholipase C. Okay, so you've got whatever it is, your ligand's going to bind there. Okay, it's then going to turn on phospholipase C, which phospholipase C is then going to activate IP3. And IP3 is going to go and it's going to cause calcium release from the SR, or not the SR, from the ER, from the endoplasmic reticulum. Then that calcium from inside instead of outside is then going to bind, and you're going to release acetylcholine, or sorry, glutamate, I think is what they said it was. You're going to release some kind of neurotransmitter, which is then going to go back into the brain, and based on the neurotransmitter, based on whether it was salt, bitter, or umami, is going to tell your brain what you're eating. Okay, so that's how taste works. Then you've got your smell. Okay, so you've got these kind of dendritic-like neurons. Okay, and you have a G protein coupled receptor on here. Okay, G protein coupled receptor. This one is adenylate cyclase. Okay, so you've got some ligand. It's going to bind to that. Then this is going to go over there and it's going to activate adenylyl cyclase, which is then going to make cyclic AMP. And this cyclic AMP is then going to open sodium or potassium channels to then let your smell come in. It's going to send a BWONG, which is then going to go up to your brain and it's going to say, hey, this is what we smell. And apparently we've got lots of different smellers, so that's cool. Let's go ahead and do some eyes now. Yeah, let's do eyes. Okay, so for your eyes, let me draw first. Alright, you get the gist, I think. Okay. Yeah, anyways, you get what I'm going for. Okay, so you've got this thing that comes over like this. This thing right here would then be considered like your pupil. Okay, then you've got these ciliary ligaments that are going to come, or not ciliary, sorry, suspensory ligaments that are going to bind to your lens. Okay, and then down here, we'll make it red. This is where your ciliary muscle or your ciliary bodies are. Okay, so when these contract, your lens becomes more circular. Oh, hang on, let me rephrase that. When these contract, you get slack in the ligaments, which then make your lens circular. Okay, and then behind all of this, you've got your photoreceptor, your bipolar body, and then your neuron that's going to send it back to your brain. Okay, so the role of this lens is going to be to focus light, right? So based on where the light is, it has to focus it either less or focus it more. So let's say that we have green light, okay, and the light's coming from way back there. Okay, so it's going to go to there, and it's going to hit your lens, and then the lens is then going to focus it back like that. Okay, but then let's say you have closer light, do this purple, you've got light that's closer, right, it's going like that, and then still like that. So things that are closer, you can kind of see this angle, it has to bend it more, whereas if it's further away, you have to bend it less. So there's a couple different things that you can use to fix if you have some kind of eye problems. You've got a lens that could look like this or like this. These are like glasses. This is your concave and this is your convex. So let's say that your ciliary muscles were not working. Okay, if your ciliary muscles could not contract and make the lens circular, that means that the lens would be flat. Okay, if the lens is flat, that means that you don't have to bend light as much, right? A more circular lens means you bend the light more. A flatter lens means you bend the light less. So this one is not bending light very good. Okay, so a flat lens is not bending the light very good. So you need something that's going to bend the light more, which is a convex one. It's going to hit it like this and then bend it more. A concave one is going to hit it and it's going to spread it out. So if your eyeball was like growing back here and that's where it needed to hit instead of right here, right, you would use a concave because then it would spread it out. It would spread it first so that it could then reach all the way back there. So that's how that vision works. Okay. Let's go ahead and do hearing and then I'll save another video. So for hearing, you've got your cute little ear. And that's going to be your duct. And then you've got your bones. Look like that. That's kind of ugly. Okay, so you've got your sound waves coming through here. Blah, 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 blah. It's going to then go through, this is your auditory duct. It's going to go through your bones, your stapes and stuff. Okay. And then over along here, this is your basilar membrane. It's going to go like this. And so it's going to kind of shake this. And then if it matches that frequency, it's going to send a neuron signal to the brain and tell you, hey, like that's what you're hearing. So we'll do a cross section of that real quick. Okay, so you've got circle, that's your ear. Okay, this is that top section, that's that bottomish section. You've got what's called your tectonic plate. This is the basilar membrane I was talking about down here. And then you've got your hair follicles that are going to look like that and go up in like that. Okay, so there's an area over here. Let's move this over there. Okay, so there's some cells over here, right there. Okay, and the purpose of them is to increase potassium. So potassium in here is very high. And that's because of these cells. You've got something called your NCK. It's going to bring potassium, sodium, and chlorine in, right? And then the potassium is going to leak through here, and then you got more NNCK channels. It's going to do the same thing, potassium, sodium, chlorine. And then you're going to have more potassium here, and it's going to keep going all the way until you get high potassium in there. Okay, the chlorine leaks back out here. All right, so then what happens is as you are hearing, as frequencies go through, you move these ciliary muscles. Okay, so you're going to have ciliary muscle. Let's do a different drawing. Okay, so you've got these ciliary hairs that look like this. Okay, and you have channels that are connected like that. These are potassium channels. But when you hear and it moves the hairs, right, it's going to cause that to open. Do you see how that happened, right? It stretched, so it pulled that springy thingy, so it opened. Okay, so now you've got it like this, and you've got ciliary, whatever. And then you have some vesicles with some neurotransmitters. And you've got the brain. Okay, so that opens, so you get potassium coming in here. Inside here, the charge is like -40. Out here, it's -80. So in total, you have -120 as your driving force for your potassium. So it opens, potassium is going to rush in, and then there's a leak channel that's going to let it go out eventually, but not before it depolarizes the cell and you get calcium in to then release these neurotransmitters. Okay, and it just depends on where on this do you hear it. Does the frequency match, right? Because it's going to kind of reverberate, and then it's going to be like, oh, hey, I matched this frequency right here, so then that's the neuron that it's going to send. Okay, and if you're looking at our basal membrane, right, the closer to here, I guess I'll do it up here, the closer to this is the higher frequency, then the further away is the lower frequency. It kind of rolls out like that. Okay, so that's how your hearing, taste, and smell work.